The Agronomy and Horticulture Study Program have a vision to become a center of academic activities with international standards in crop production technologies in a sustainable tropical agricultural system. Agronomy and Horticulture is an undergraduate study program dedicated to produce human resources in the field of agronomy and horticulture, which focuses on principle of crop production, crop ecophysiology, plant breeding, biotechnology, and seed science. We prepare graduates for an early professional career in agriculture-related industry, public and private sectors, as well as to postgraduate studies. The Agronomy and Horticulture Study Program has 450 students, with a very competitive enrollment all over nation. You will ensure personal guidance throughout the student career with a student-teacher ratio of 6:1. Agronomy and Horticulture Study Program has a total 71 academic staff members graduated from reputable universities all over the world. Agronomy and Horticulture Study Program offer you excellent facilities. We have 11 classrooms, library, executive lounge, toilet in each level, and praying room. For accommodated study and research process, we have 26 laboratories, including molecular biology laboratory, plant breeding laboratory, testing laboratory, ecotoxicology and bioagent laboratory, post-harvest and biomass laboratory, tissue culture laboratory, seed testing and storage laboratory, microtechnic laboratory, and 70-hectare experimental fields, including Lewi Kopo and Cikarawang Experimental Field for lowland research, Sawah Baru Experimental Field for paddy research, Cikabayan Experimental Field for plantation research, Jonggol Teaching Farm for palm oil research, Sukamantri Teaching Farm for fruits research, and Pasir Sarongge Experimental Field for highland research.
The Agronomy and Horticulture Study Program always prioritizes safety in all activities. Personal protective equipment, clothing for practicum when applying pesticides in the garden, and shower laboratory are some of the safety facilities for the Agronomy and Horticulture Study Program. To anticipate a fire, the Agronomy and Horticulture Study Program has been prepared smoke detector, alarm, and evacuation routes. Fire extinguisher and hydrants has been prepared at each classes, each laboratories, and another places in accessible locations. In addition, a fire engine is stationed on campus in case of an emergency. A first aid kit is kept in each classroom and laboratories, just in case of injury. If students in critical condition, there is a clinic on the IPB campus that open 24 hours a day. This clinic is provides inpatient care and an ambulance for bring the patient to the nearest hospital. To prevent the spread of COVID-19, health protocols such as social distance and hand sanitizers are placed at every classroom, laboratory's door, and at the wall, where there are many potential people doing activities. Furthermore, sinks are placed at campus or field for washing hands.
Academic program based in Agronomy and Horticulture Study Program all has been certified for A-level from National Board of Accreditation for Higher Education and ASEAN University Network Quality Assurance 2013 to 2017. The academic administration and learning process have been certified by Sukuvindo International SNI ISO 9001. Academic atmosphere at Agronomy and Horticulture Study Program is very conducive. For education and research, we collaborate theory and practice in the curriculum. Domestic students have the opportunities to participate in student mobility or student exchange program to other universities, such as ASEAN International Mobility for Students, Swiji Service Learning Program, Summer or Winter Course, International Internship to Netherlands. Agronomy and Horticulture Study Program fully support non-academic activities for students to enhance soft skills, leadership, and entrepreneurship, such as student association, sport and art, international and national competition, festival, and others. Research activities at Agronomy and Horticulture Study Program is very intensive. We have produced new crop varieties and production technologies in addition to scientific papers and awards. Agronomy and Horticulture Study Program has reached its collaboration with many national and international research institutions.
Selama di program studi PBT, saya banyak belajar tidak hanya terkait pemuliaan konvensional, tetapi juga jalur bioteknologi dan juga mutasi, dan juga manipulasi genetiknya.
Saat ini saya sedang meneliti tentang kelapa, khususnya di bidang molekuler. Saat ini saya menggunakan marka SSR dan SNFI melalui gen pertambahan tinggi tanaman. Motivasi saya memasuki program studi ini adalah untuk mengeksplor lebih dalam lagi keragaman tanaman kelapa. Oleh karena itu saya melakukan penelitian di bidang molekuler ini.
Motivasi saya memasuki program studi pemuliaan dan bioteknologi tanaman, di mana saya ingin mengeksplor diri dan mendapatkan pengalaman dari dosen-dosen pengajar yang profesional di bidangnya.
Motivasi saya memasuki program studi pemuliaan dan bioteknologi tanaman adalah karena fasilitas yang ada di IPB ini sangat lengkap.
Motivasi saya mengambil program ini adalah saya ingin menciptakan varietas tanaman yang bermanfaat bagi umat manusia.
Saat ini saya sudah memasuki semester 2 dan sedang melakukan penggaluran tanaman jagung. Alasan saya masuk jurusan program studi ini adalah saya ingin menjadi breeder yang handal. Dan dosen-dosen di program studi pemuliaan dan bioteknologi sangat unggul dan kompeten.
Yang menarik dari program studi ini adalah saya dan teman-teman bisa belajar tentang pemuliaan secara konvensional dan juga bioteknologi dari dosen-dosen yang handal, serta telah menghasilkan inovasi-inovasi yang baru dan mutakhir dalam bidangnya.
Riset-riset yang dilakukan di program studi pemuliaan dan bioteknologi tanaman ini difasilitasi dengan fasilitas yang cukup memadai serta lengkap. Oleh karena itu, penelitian di bidang ini dapat berjalan dengan lancar serta menghasilkan inovasi-inovasi yang mutakhir.
Penyelenggaraan proses studi pada program studi pemuliaan dan bioteknologi tanaman didukung oleh para dosen yang sangat berkualitas, prasarana pembelajaran yang sangat baik, kebun percobaan dan laboratorium yang terkelola dengan sangat baik, serta atmosfer akademik yang sangat kondusif.
Di program studi pemuliaan dan bioteknologi tanaman ini, kami menempa anak bangsa untuk menjadi pemulia tanaman yang handal melalui pendekatan pemuliaan konvensional maupun bioteknologi di tingkat seluler maupun molekuler. Indonesia membutuhkan SDM yang handal di bidang pemuliaan dan bioteknologi tanaman. Karena itu, melalui program studi pemuliaan dan bioteknologi tanaman Sekolah Pasca Sarjana IPB, kami siap untuk mendidik anak bangsa.
Program studi pemuliaan dan bioteknologi tanaman mendidik calon-calon pemulia tangguh yang mempunyai latar belakang ilmu yang kuat dan pengalaman yang mumpuni dalam bidang pemuliaan dan bioteknologi tanaman, baik pengalaman di lapangan maupun di laboratorium. Oleh karena itu, mari bergabung bersama kami di program studi ini.
Mari bergabung bersama kami, studi lanjut di program studi pemuliaan dan bioteknologi tanaman Sekolah Pasca Sarjana Institut Pertanian Bogor.
Ayo, mari bergabung di program studi pemuliaan dan bioteknologi tanaman.
Ayo bergabung bersama kami di program studi pemuliaan dan bioteknologi tanaman.
Mari bergabung di program studi pemuliaan dan bioteknologi tanaman.
Ayo bergabung bersama kami di program studi pemuliaan dan bioteknologi tanaman.
Mari bergabung di program studi pemuliaan dan bioteknologi tanaman.
Mari bergabung di program studi pemuliaan dan bioteknologi tanaman.
Mari teman-teman, bersama kita bergabung pada program studi pemuliaan dan bioteknologi tanaman Sekolah Pasca Sarjana IPB.
Palm oil is an edible vegetable oil that is in nearly half of packaged goods in supermarkets, as well as in animal feed, biofuel and cosmetic products. Due to its high demand, farmers are under pressure to increase yields in agricultural production, which risks detrimental impact on the environment. In order to improve production sustainability and meet growing demand, palm oil producers are turning to genomic selection and marker-assisted breeding to preferentially select for palms at an early stage of development that lead to higher yields when they mature. Our customers have identified functionally relevant markers linked to their desired traits. These markers can be tested using LGC Biosearch Technologies products such as CASP and SNPline, which automate the genotyping of seedlings at high throughput. Overall, this approach generates millions of data points monthly and has significant impact on the productivity of the plantations. We partner with palm oil producers to sustainably improve farming productivity through marker-assisted breeding. We provide our customers integrated genomic analysis tools that accelerate their mission-critical applications. LGC, science for a safer world.
Ladies and gentlemen, we also would like to express our sincere gratitude to Head of Department of Agronomy and Horticulture, Professor Edi Santosa, Head of Indonesian Horticulture Society, Professor Dewi Sukma, our honorable speakers, Professor Darda Effendi, Bredihan, and also Darsna Fias. And also our moderator, Dr. Sinto Wahyuning Ardi. And last but not least, our dearest participant, thank you for joining this event. It's been a great honor for me, DKP Tapsari, to be here and welcome each and every one of you in this meeting. Over the next 3 hours, this meeting will provide you a plenty of insight and information on the role of NGS-based technology in biomarker development, particularly in biomarkers utilized in agrigenomics, and also introducing the workflow solution from LGC Biosearch Technologies for agrigenomics applications. Well, before we proceed to the program today, let me hear the welcome speech of the program from the Head of Agronomy and Horticulture Department, Faculty of Agriculture, IPB University, Professor Dr. Edi Santosa. To Prof. Edi, the time is yours.
Okay, thank you very much, Ibu Dika. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Morning. Welcome for Ibu. We call Madam in Indonesia call Ibu Dasti, Ibu Dasti, and welcome also for Mr. in Indonesia call Bapak. Bapak Brady. Thank you very much for Madam Dusty flight from London to Indonesia to see all of students in this study program. Okay, my name is Eddie Santosa. Just call me Eddie, Head of Department of Agronomy and Horticulture. And thank you very much for actually Ibu Dewi, Professor Dewi, is a president of Indonesian Horticulture Association. And she's also the chairman of the study program, plant breeding and biotechnology. Without her, I think this event will, how to say, far from our expectation. Okay, give applause to Ibu Dewi. And also our young lady, Bu Sinto. Thank you very much, Bu Sinto. And also Pak Profesor Darda Effendi. Thank you, Pak Darda. Okay, I have very short speech actually. Biotechnology will be our new frontier. I think this word, how to say, summarize all. And Madam Dusty, I think she will deliver about how to explore the molecular breeding using genetic selection and associate technologies. I think it's very interesting. And also Pak Brady will share about the idea how nucleic acid purification technology from LGC. Thank you very much again for the attendance online as well as the offline here. And I hope this half-day seminar will be a benefit for us. And I think thank you again for Ibu Dusty and Pak Brady for coming here. And also Professor Darda as a moderator. Not moderator, speaker. Sorry. Because usually on the list, top part is moderator, but actually, okay, speakers. And moderator will be Ibu Sinto. Thank you very much. And thank you Biosearch and Technology for as a sponsor of this program. Okay, please enjoy. Assalamualaikum warahmatullahi wabarakatuh.
Yeah, thank you, Prof. Eddie, for the welcome speech. And we are now going to hear also the welcome speech, but this time we will hear from the head of Indonesian Horticulture Society, Professor Dewi Sukma. Prof. Dewi, the time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. Honorable the head of Department of Agronomy and Horticulture, Professor Eddie Santosa, and our great speaker, Madam Dusty. Sorry if I make a wrong to pronounce your name. And Professor Darda Effendi, our professor at Agronomy and Horticulture Department. Lady, thank you for coming, Annie, and also the team from Science. Thank you very much for your to support this event, and specifically for the team of the committee, Dr. Sinto and our students. They already work very hard to make this event possible. Please applause for them. And also, thank you very much for all of our students. You may see in this room, all of them are our students. So we have a lot of students for master degree and also PhD students. And Thank you also for the students and maybe researcher from other institute or lecturer from other university that can join in Zoom. Welcome for this event. I think this is a very important event. I'm happy to have it because, as we know, we have the very high diversity for horticultural crops, especially for fruits. And however, there is limited varieties that we can develop on fruits because of the complexity of genetics. And the fruits have very long juvenile stage, and if we cross, we need a long of time to see the result of the new varieties. That's why we need the molecular approach to help this to help the building of the fruit crops. So I think this the material that you share, the speaker shared today will enlighten our knowledge. They will share their experience, knowledge, and also many things that can be absorbed and help us to develop the roadmap of our research in the future, so we can improve our plant varieties, specifically on the horticulture crops or fruit crops. That's all from me. Thank you very much. I apologize for inconvenient things in this event. I hope we will get a lot of benefit from this seminar. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Prof. Dewi, for your welcome speech. So, the honorable participants, I hope you had fun until this time. And then we are ready to follow amazing talks from our amazing speakers. And to start the first session, maybe let me briefly introduce our moderator who will lead the session. So the moderator will be Dr. Sinto Wahyuning Ardi. Dr. Sinto is a lecturer in Plant Breeding and Biotechnology Study Program, Department of Agronomy and Horticulture, IPB University. And she earned her bachelor and master degree from IPB and obtained her PhD from the University of Tokyo, Japan. Dr. Sinto research focus on plant molecular biology and plant tolerance mechanism to abiotic stress, particularly in foxtail millet, Setaria italica, and other minor crops. So, ladies and gentlemen, please welcome Dr. Sinto Wahyuning Ardi. The time is yours.
Terima kasih Pak Kadep atas waktunya. Semoga nanti bisa bergabung kembali. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning. Selamat pagi. It's my great honor to be here to moderate the sessions. We are going to have a very experienced speakers in biotechnology area, and I will not sit before I invite them. So I will happily invite our speakers of today, Dusty, if I may call you that way. So our speaker from LGC Research and Technology, Madam Dusty first, and please come to the front. Honorable participants, and I would like to inform that today approximately 200 participants are joining online and on site because of the room limitation. That's what we have here to present, but many are joining online. And our topic today is the role of NGS, next generation sequencing technologies, in the development and analysis of biomarkers for Agrigenomics. It's a leading and emerging issues. So the first speaker from our department, Professor Dr. Terda Effendi, will start the session. So I would like to introduce his profile briefly. So Professor Darda Effendi, I call him Pak Darda every day. We are from the same division. He's a full professor in the Plant Biotechnology Division of Department of Agronomy and Horticulture. And his research interest is focused on horticultural crops, mainly fruit crops, using both tissue culture or cellular biotechnologies and also molecular technologies. He's been the secretary of the plant breeding and biotechnology study program, and also he's become the director of the Center for Tropical Horticultural Studies at IPB University until 2018, and currently he's the secretary of the doctoral program of plant breeding and biotechnology in our department. And he is the member of our IPB Academic Senate. So today he will talk about application of molecular biotechnology on tropical horticultural crops. And I would like to mention briefly about his outfit today. His outfit today is, I'm sorry, but his outfit today is batik with a molecular pattern. Yeah, so because he is a full professor in biotechnology. So Professor Dada, without further ado, please share your lecture in maximum 40 minutes. The time is yours.
Thank you, Ibu Sinto. Assalamualaikum warahmatullahi wabarakatuh. And welcomes everybody to this meeting. Actually, I'm a little bit sleepy, but it's okay. This time's efficiently, I just start directly of the topics. The topics today is application of molecular biotechnology on tropical horticultural crops. Some is theoretical and some is what we do, what we done in our labs. Let's continue. Next. Okay. So the topics for today is molecular biotechnology, the definitions, then horticulture and its complexities, and then the challenge of application of molecular biotechnology in tropical horticulture crops, application of molecular markers in tropical crops, and then case studies of what we've done, and then genetic transformation of horticulture crops, and the relationship between cellular biotechnology and molecular biotechnology, and also the roles of next generation sequencing based technologies on tropical horticulture crops, if we have enough time. Next. Let's start with what's the popular about molecular biotechnology. It's some definitions, but yeah, maybe the next speakers will talk more about what biotechnology is. It's a multidisciplinary field that combines principles of molecular biology, genetics, biochemistry, and microbiology to develop technologies and products for various applications, particularly in agriculture, medicine, and industry. In scientific definition, molecular biotechnology is the use of laboratory technique to study and manipulate the structure and function of the molecular, particularly DNA, RNA, and proteins, to understand biological process and to develop practical application, for example, to breed the new variety or the new genotypes. And for the agriculture, of course, the biotechnology involves the application of genetic engineering, genome editing, and other molecular tools to improve crops, plants, and livestock also, and for better life of human being. Next. And this is many topics that includes in molecular biotechnology. See, from the molecular markers, marker-assisted breeding, marker-assisted selection, and plant genomic and functional genomics, and transformation, genetic engineering, and then the last one, genome editing or gene editing, prime editing, and base editing, something like that, and then metabolic engineering, plant microbe interactions, and epigenetic and gene regulations, and go to the plant synthetics biology and many more topics in plant molecular biology that can be done. And in the other side, we are talking about the horticultures and complexities, difference from the food crops. The horticulture is more complex because of some, including biological complexities, genetic diversity, and perennial in nature, and also reproductive biology of the horticulture is very varies in model. And for examples, there is cross-pollination, self-pollination, self-incompatibility, and once plants like papaya have 3 kind of sex, and then also most of vegetatively propagated like mangosteens. There is apomictic plants that cannot breed by the crossing because there is no father in mangosteens, just mother. Also in banana, commercial banana is almost seedless. So the way to go for the crop improvement just by the breeding, biotechnology-based crop improvement. And also horticulture's maturity is much different than just physiological maturity. We were talking about that. And then also environmental challenge, climate sensitivity, pest and disease, and also soil health, and many more problems in horticulture. Next. In Indonesia, for example, we, based on the Ministry of Agriculture's decree, horticulture consists of fruits, veggies, vegetables, herb or medicinal crops, and flower and ornamentals. We have 60 fruit species, 82 veggies, 220 flower or ornamentals, and 60 species of herb or medicinal crops. But each species consists of many clone or variety. For example, durian maybe have 100 or thousands different genotype. So the variation is very high. So it consists of diverse species of great economic potential values for food, body and soul, functional food, healthcare, cash, crop and national economy, industrial development, and other for environmental protections. Okay, next. Just go through. Next. Just click OK. So this is also one of the problem in horticulture. Horticulture is harvested since the sprouts until almost senescence. So if we are breed for microgreen, for example, is different. We are breed for the mature fruits. So many difference in what's development stage of harvesting of horticulture crops. So everything needs specific treatments. Okay, next. Okay, the challenge of application the molecular biotechnology in tropical horticultures, because the complexities that we are talking about, genetic complexity in tropical horticultures. Many tropical horticulture crops such as bananas, mangoes, papayas have complex genomes. As papaya, for example, also have triple sex, male, female, and hermaphrodites. And then most of the consumer just prefer the hermaphrodites fruits, except some countries produce female fruits. Okay, so also says already talking about the mangosteens, we are impossible to do the conventional breeding except for maybe mutation induction or poor man breeding, they said. And so it needs to go to the biotechnology. So because the complexity including polyploidy, high heterozygosity, it makes genome sequencing and other thing is complicated. It's much difficult than foxtail milk and then rice and other things. Okay. And also lack of genomic resources compared to the temperate crops. Many tropical crops lack wells annotated because most of the tropical crop is just like say orphan crops. Yeah. Slow down research and development efforts, limit the use of tools because mostly tropical countries is developing or let's say poor countries. So biotechnology is at least in the beginning is very expensive. Okay, let's next. And also some other factor, environmental stressors, and perishability and post-harvest losses. You know that's the first commercial GMO is tomato from Calgens or something that manipulates the tomato for better post-harvest life. We are talking about that later on. And we are already talking about the complexity of reproductive systems. Some is vegetatively propagated. That's impossible to do the crossing. What need cellular biotechnology and of course also molecular biotechnology, because it's impossible to do hybridization. Maybe somatic hybridization, but it need also cellular and biotechnology. Just continue. Okay, and we also a lots of pest and disease and dense ethical and biodiversity concerns about the GMO. Yeah, some consumers still don't want to eat anything that's biotechnology-based breeding. Yeah, even the CRISPR case, some just still think that is GMO. That's a problem. And also socioeconomic factors. Next slide, please. And this is some application of molecular markers in tropical horticultures, marker-assisted selection and marker-assisted breedings, genetic diversity analysis, quantitative loci mapping, germplasm characterization, paternity testing and pedigree analysis, disease and pest resistance, and symbiotic stress tolerance. We have some examples that we are doing in marker-assisted selection or genetic diversity analysis. Next, please. And let's see some developments of markers since era before the PCR. The restriction length polymorphisms with DNA-based markers that's not based on the PCR. After this is first DNA-based markers by restriction enzymes and what's after, I think in 1986, we have PCR machine, thermocycler. Then after that develops lots of markers based on the PCR. The first one is maybe the RRPD. rapid screening of genetic diversity, but because the RIPD use very short primers and is not really reproducible. Okay. And then develops SSR, that's key for marker assisted selection, and enable also QTR mapping for complex traits, and it might be codominant in nature. And also lately, since 19th and early 20s, developed SNP, SNP, and then develops more and more next generation sequencing based markers, and also then CRISPR based markers, and then in the future might be single cell genomic, or the hot issue also how the epigenetic something's done to the traits or new traits, so how epigenetics markers can be useful in the future, and then maybe AI driven markers discovery. So from the non-PCR to single cells, from restriction enzyme based markers to more easier and can be automatized. Okay. What we in the 90s still working a lot in RIPD. Actually, until now, we still some have RIPD and also Scott's markers. Yeah, our student doing RIPD and Scott markers, SCOT markers. So, seems time is not too many for me. Let's continue. This case studies some of our what we are doing. This is mining and characterization of genomic-based microsatellite markers in Duku, Lansium domesticum. We are looking for SSR markers, and we find some microsatellites with motif hexanucleotides, pentanucleotides, tetra, trinucleotides, dinucleotides, and mononucleotides, and mostly trinucleotides repeating. Okay. Next slide. And then we also working for the same as Lasium domesticum or synonym to Lasium parasiticum. We would like to know if what's a vernacular name, because the same vernacular names may be for different species or different variety or different genotypes, and then maybe different vernacular names is done for one specific species. So in this case, we are Lansium domesticum, some called Duku, some called Kokosan, some is called Langsat. So we would like to know if Duku, Kokosan, Langsat, and Duku, Langsat, Kokosan have the same genotype, and we are looking for several origin of this Duku or Lansium species. One from the Chondets in Jakarta area, and then it's in Mekarsari fruits, fruit park, and then from the Bogor Botanical Gardens. And from the 12 SSR markers, we can differentiate between Chondet Duku, but in some groups they still consist of duku, langsat, and kokosan. So cannot differentiate the things that from the mekarsari. Maybe, yeah, maybe they are little bit different. So I think the 12 SSR primers that we use is some working to differentiate with Chondet Doku, and but not for others accession. So still things to do for the next research. Next one, please. And this we are working also to characterize papaya ring spot virus, because papaya ring spot virus is also already spreads in Indonesia, especially in Java. And then we are looking for several location in Java, in Bogor, Bogor 1, Bogor 2, Bantul, Purworejo, Kebumen, and then we check with the control zucchini yellow virus, yeah, I think. And then we find that all of Java accession of papaya ring spot virus has high similarity. It's also high similarity with the one from the Thailand, but it's different quite a lot from others from the Bali, Nganjuk, Vietnam, China, Brazil, and Australia. So maybe there is in Java more than one genotype of papaya ring spot virus. Okay, just continue. Next one. This we are also working on sweet potato. Isolation and characterization of cDNA clones encoding novel subfamily sporamin B in sweet potato. This is genes that have effects on what we call its skinning injuries. Yeah, skinning injuries. Because during the harvesting, maybe the sweet potatoes get injured by the process of the harvesting. So some genes differentially expressed during 2, 4, 8, actually 8 and 12 hours after we give the treatments of skinning injury. And so different genes with different times has different size of bands. This is a difference Apomia batatas H41, 23, and 74. There's 3 different genes related to sporamins B in sweet potato. Next, we are using ACP annealing control primers. This is that's 3 genes that skin up regulated and then skin transient regulated. The upregulated increase from control to 2, 4, and 8 hours after the treatments of skin injury. Then for the IBS41 is skin transients regulated at 2 hours and go down to 4 and next 8 hours. Okay. Okay, let's continue. Just so. Okay. And also we find some others differentially expressed genes during the skinning process, skinning injury process, including for the abiotic stress, lignin, and severins, and regulation and signaling. Some is in use, some is up-regulated. Just one is down-regulated in here for the regulation and signaling. So maybe just for the skinning injury process, a lot of genes have differentially expressed during that times. Maybe for wounds recovery, something. The next. And this we are looking for genetic stability in tissue culture of sugar canes. We are using 1, 2, 3, 4, 5, 6 clones of sugar canes, and we compare the stability of tissue culture from what's subculture, the third subculture to the 6 subcultures to the 9 subcultures, and the SSR markers groups the clones correctly, and then most clone is have very high similarity, except for one clone, the lowest one, the PS862, is the other is similarity at 94%, but the might be accumulation of somaclonal variation in the clones of PS892, 62, quite more than other clones. So the similarity is just about 64% or 0.64. So it proves that genetic stability in tissue cultures is genotype dependent. Depend on the genotype, depend on the genetic makeup of the clones. Some clone is stable from control, the mother plants, to the 6 subculture. But the last clone, the PS8622, has quite difference between mother plants, mother explants, third, 6, and 9 subculture. So prolonged cultures in tissue cultures will increase the possibility of mutation, or what we call is somaclonal variation. Goes, next one. And let's move to genetic transformation on horticulture crops. It's much different than food crops. For fruit crops, the company doing the GMO for the herbicide tolerance, insect tolerance, and others tolerance, especially for seeds company doing things. But for the horticulture crop, we are working mostly to improve shelf life, because all of the horticulture is very perishables. The post-harvest loss might be from 20 to 50%. So it's a lot of losing in in horticulture, especially from vegetables and fruits. So scientists try to improve the shelf life, or prolong the shelf life, or the post-harvest life of the horticulture crops. And then, of course, increase yield and productivity, improve quality, nutritional quality, or just the visual quality, appearance. Appearance is most important in horticulture. Some also enhance stress tolerance and reduce environmental impacts of the commodities. Next one. So this is maybe the simplest timeline of genetically modified horticulture crop. The first one, the commercial GMO, the first commercial GMO, it's Clavosaphor tomato. That's try to prolong the shelf life of tomato. We are talking about this later. And then also new leaf potato, BT, Bacillus thuringiensis is also one of technique to improve resistance to the insects. And then one good story about the rainbow papaya from the Hawaiian papaya that's can resistant to the risk virus. But rainbow papaya also sad story after Hawaiian papaya gets attacked by ring spot virus, and then scientists try to make resistant papaya to ring spot virus, and they got the new variety. But still that era in 90s, a lot of consumer very contra to the GMO. So Hawaiian papaya cannot be sold to Japan because the first market for Hawaiian papayas is Japan. So Japan cannot accept the GMO at that time. So it's sort of problem. So some squash resistant to zucchini yellow mosaic virus, watermelon virus, sweet corn with the BT traits for insect resistance, and finally, lately, Arctic apples resistant to browning and when cuts or blues, and then also develops just want to change the colors of pineapples from orange or yellow pineapples to pink flesh pineapples. Maybe pink pineapples looks better. Just looks different. Looks better. And then also the technology also develops from genetic engineering based on transgenesis, and then develops to genome editing, and lately to the CRISPR edited tomatoes, and maybe next time also just prime editing or base editing. And luckily, CRISPR edited tomato is approved for commercial sale in Japan with higher levels of gamma aminobutyric acids. This is considered is not transgenic, it's not GMO, because it's just to edit the genes, the endogen genes that naturally belongs to the tomato. So no DNA from outside put in the GABA tomatoes. So lots of horticultures, vegetables and fruits develops for just quality, and but not many big company working for the horticultures, mostly working for food crops, right? Because the market is it's just different. This just niche market for horticulture crops. Continue, please. Okay, let's say this is the first commercial GMO, the Clavosaphor tomato. That's traditionally tomato is harvesting green nature, and then after distribution they treat with the ethylens to rapidly mature or change the colors. And then scientists make a flavor saver tomato that can be harvesting ripe because they use the antisense genes of polygalacturonase to block the gene expression of polygalacturonase that actually polygalacturonase will play roles in softening of the fruits. So if the gene is silenced by in that technology, it's 90s technology. The way to silence the gene by introduce the same genes in the antisense orientation. But now the new technology, we can just edit what the genes and let's cut the genes in the specific targeted area. So this is approved and then marketed under McGregor's. But during that time, even in America, USA, the market or the consumer still not really happy with the GMO. So the supporter of tomato claims that the company requires specialized transportation equipment and post-harvest facilities differentiate between normal tomato and GMO tomato, but it's impossible in terms of economy. So the flavor saver tomato is get pulled from the market. That's the story of flavor saver, the first commercial GMO that try to give better post-harvest life of the tomato. Just next slide, please. And then more than 20 years ago, I worked also for the metabolic science strategy to reduce ethylene biosynthesis by the precursor of ethylenes by hydrolyze the S-adenosyl methionines to the methionines, so it's not used to synthesize the ethylenes. Thereby using same hydrolyze or same K in this same K plasmid, harbor plasmid. But I did for the avocado, but needs a long time to get because the genes, the same case controls by specific promoters that just express during fruits just before the maturity. So it's not constitutive promoters. You cannot recheck the expression during the tissue cultures process. Next, please. And then this is the new one, the pink, yeah, they are doing commercial known as Del Monte Pink Glow pineapples. The genetic modification goes involve the knockdowns, also the silencing, or at least knockdowns the lycopene beta cyclase genes, causing lycopene or red pigment to accumulate, not to go to the next reaction, become the yellow one. Okay, and then this is GABA that is I think is already in the market for the high aminobutyric acids. That's better nutrients for us, for the consumers. And then for the lots of commercial, you can check in the ISAAA website many fruits, flowers, and vegetables that's it's already some already in the market, but some still. Yeah, papaya is maybe the second one most popular commercial GMO of horticultures. Next slide, please. And then since we are also working on cellular biotechnology, so what's the relationship of cellular biotechnology and molecular biotechnology? The question, can molecular biotechnology-based crop improvement stands alone? I think we thought the tissue culture. Or more specific questions is, can molecular biotechnology-based crop improvement of breeding bypass tissue cultures protocol? A lot of scientists try, yeah. Or can transgenesis and genome editing-based crop improvement bypass tissue cultures protocols? Yeah, the answers can be yes or no. Yeah, yes for model plants, the small plants like Arabidopsis and other small plants. But for the tropicals, especially woody perennial crops like mangosteen, mango, banana, it's difficult to do without the tissue cultures. So let's see the next one, the next slides. So cellular biotechnology and molecular biotechnology have support each other. From a cellular biotechnology or tissue cultures, say simple tissue cultures, we are working on pre-existing meristem cultures, organogenesis, somatic embryogenesis, protoplast cultures, haploid cultures, cryopreservation or in vitro conservation, and many more. And then in molecular biotechnology, we identify, isolate, cloning, gene expression, and genes engineering, genes transformation, marker-assisted breeding, metabolic engineering, for examples. And then for the genetic engineering or genome editing or other precision breeding based on molecular biotechnology, we need, of course, tissue culture respondations as technique for application of or how to deliver the genes or the CRISPR reagents or the transformation or transgenes from the outside to inside the cells. It's not possible yet. We do it in the in-text plants. So we're still doing it mostly on the somatic embryogenesis or somatic embryo. So delivery construct reagents of either genetic engineering or genetic transformation or genome editing, still the bottlenecks for process. So still needs the tissue culture. And then after we get the new genotype, for example, by the molecular biotechnology, we need also tissue culture for recovery and rapid propagations, and then also in vitro selections, marker-assisted selections, in vitro conservation or cryopreservation or the new genotypes. Okay, so especially for the tropical horticultures, the molecular biotechnology still needs support from the cellular biotechnology. Impossible the molecular biotechnology stand alone, except for just marker assisted selection on the some vegetables or genetic engineering based on the pollens or seeds. Okay, but other than that, especially for perennial crop, we still need the tissue culture. Next one, please. this what we can do with the roles of next generation sequencing based technologies on tropical horticultural crops, maybe for genome sequencing, assemblies, identification of genetic markers, especially SNPs and others specific markers. Okay, next one, just continue. And transcriptomic and gene expression analysis for our commodities, and then epigenetic and DNA methylation. Epigenetics now is a hot issue also in biotechnology, because epigenetic change the traits, the characters, without changing the DNA sequence or gene sequencing. And then meganomics and microbiomes analysis, and then also pathogen detection and diagnostic, or how interaction between pathogens and our plants, and also comparative genomics. Then maybe NGS will be more issue from the Brady and okay. Next one, maybe this last one. Yeah, functional genomic reading and trait improvements, discovery of novel new genes and alleles, and genomic editing and CRISPR-Cas9 applications. Of course, to get the, for example, the RNA guides, we need to know the sequence, and also to check the PAM of the guides, we need to have the sequence of entirely genome, for example. It's easier for the next generation sequencing. If we are working for RAPD and ISSR, we don't need sequencing, but for this generation of technology, we need to sequence. For us, the problem is still expensive. That's one problem. Okay, next one, please. Okay, thank you, and hope you can understand my English. Thank you.
Further for the insightful talk, highlighting the complex nature of horticultural crops that necessitate the molecular biotechnology approach for the crop improvement, because some crop cannot be improved traditionally, right, professors? And most of the approach mentioned by Prof. Dada, mentioning next generation sequencing, that needs the DNA as starting materials, and high quality and high yield DNA is necessary. So we would like to know what would be the example of improved technology to obtain high yield DNA and excellent quality of DNA. And that's why we would like to welcome Brady. Before he starts his session, let's get to know who is he. Okay. And this is his a brief CV. Brady had, or Bapak Brady, graduated from the Molecular Biology and Biotechnology from the University of Western Australia, and he is experiencing as a senior research officer in Salah diagnostic for 3 years, and then the field application for the LGC diagnostic and genomics from 2021 until now. So Brady will deliver his talk about the nucleic acid purification technology from LGC that will provide us with the high yield and high quality of DNA. So Brady, the time is yours.
Thank you. So good morning to everyone here, and also everyone behind the screens online. Hope everybody is doing well. Right, so I'm Brody from LGC, and my role at LGC is a field application scientist based in the APAC region. And personally, I'm based in Singapore. Right, so it's a pleasure to be here today at IPB University, and I'll be talking about an exciting new innovation we at LGC have developed, which is the SpeedX Lightning. So hopefully, by the end of this session, it will change how you traditionally view DNA purification methods. Next slide, please. Thank you. So to start, I'd like to give a brief introduction to LGC as a company. So LGC is a leading global life science company. We provide mission critical components and solutions for high growth areas in human healthcare and also the applied markets. Right. So our work supports important fields like genomic analysis and quality assurance applications. And we focus a lot on delivering high quality products to our customers. So our purpose in LGC is science for a safer world, right? So everything we do is driven by this goal, helping to make the world a better place, better and safer place through science. Right. And in our everyday work, we are motivated by our core values here, which is our passion, curiosity, integrity, brilliance, and respect. Next slide, please. Thank you. Right. So focusing on our genomics division, part of the business, right? We specialize in delivering essential tools for genomic analysis. Right. So we can loosely describe our work into three main areas. Right. So firstly will be the nucleic acid production. Okay, we provide high value materials like BHQ probes and primers. We also provide raw materials for oligosynthesis like our controlled pore glass and also phosphoramidite. So these are the bases used when you're synthesizing oligos. Right. So secondly, we have our molecular biology section. So we offer solutions such as the topic for my presentation today, which is the SPEEDX magnetic beads. And we also have Cas master mix for use in downstream applications such as PCR for SNP genotyping. Right. So we also have the workflow solutions. Right. This is where we deliver advanced high throughput instruments like the SNP line, our IntelliCube, the Nexa, and Mermaid. Right. All these instruments help in different capacities, which ultimately help labs improve their performance and efficiency. So all these products, they come together into a top to bottom workflow solution to support a wide range of applications from molecular diagnostics to advanced therapeutic development to molecular breeding. Next slide, please. Thank you. Right. So moving on to the topic of the day, right? It's important to clarify something we often get asked about. So what's the difference between extraction and purification? Right. To put it simply, extraction breaks open the cells to release DNA or RNA. Right. However, extraction does not remove any debris or unwanted materials. Right. So, for example, our quick extract products are designed for direct use in applications such as PCR, real-time PCR, and qPCR. So these are applications where minimal cleanup is acceptable. So it's quick and it's quick and dirty, right? So everything is fast. So I think a fun fact that Dusty reminded me this morning is that our product Quick Extract was actually brought up to space in a NASA mission. So a molecular biologist actually chose our product to bring it up to space to run experiments. So I think that's very, very cool. Okay. Right. So purification, however, goes beyond that. It removes unwanted debris and contaminants. Right. So it delivers very highly pure DNA or RNA. Right. So this is essential for more sensitive downstream applications, things like sequencing or cloning. Right. So we offer products such as Master Pure or the SPEEDX line of products specifically for these purposes. So to summarize, in short, if you need fast results for basic routine PCR or qPCR, crude extraction without any kind of purification might be enough because it's fast, it saves time, right? But if you need clean, high quality nucleic acids for more demanding downstream work, purification is the way to go, right? Purification also ensures better quality with high purity ratios. You are making it the preferred choice when accuracy and reliability are critical. Next slide, please. Thank you. So nucleic acid purification is a critical step in a lot of molecular workflows. But I think researchers often face a dilemma when choosing between crude extraction and purification methods. So let's take a look at some of their advantages and some of their drawbacks. Right. So many researchers opt for crude nucleic acid preparation due to its speed, simplicity, and cost effectiveness. Right. So it also has added benefit of using fewer hazardous chemicals, making it more environmentally friendly. However, of course, there are some downsides, right? The quality of the nucleic acids is often lower, right? So because it's a crude extraction, PCI inhibitors are present, leading to unreliable downstream applications. Right? The DNA RNA concentrations might also be a bit lower, right? And it might not be suitable for all sample types. And because of the nature of the chemistry, it might be difficult to automate if you're doing some high throughput work, right? Furthermore, quantification by UV is also challenging, so it's hard to get good estimates of your yields when you're doing your QC, right? So these limitations make crude preparation a less favorable choice for high throughput or more sensitive applications. On the other hand, traditional purification methods provide high quality DNA RNA that is free from PCI inhibitors, right? So other advantages include adaptability. So it can be adapted to nearly all sample types. It gives a higher concentration of nucleic acids. It is usually automation friendly. It has automation friendly workflows, right? And they are also quantifiable easily by UV. So it ensures accuracy in concentration measurements. Again, these benefits come at a cost, right? So usually when you're doing purification methods, the process is time consuming, especially when you're doing it manually without automation, right? It takes a lot of time. It often requires hazardous chemicals involved, right? It increases the safety concerns. And of course, these hazardous chemicals also leads to cost of waste disposal. So it's high cost when you're disposing of all these hazardous chemicals. And usually it's also more expensive compared to crude preparation, right? So automation also requires, although it's automation friendly, but it also requires a significant amount of plastic consumables. So this raises concerns about your cost, your consumable cost, and your sustainability and your waste generated. Right. So we at LGC, we try to overcome these limitations. So these are the pain points where we see from both sides of the equation, right? Both sides. We need a product that combines the best of both worlds. Right. This is where the ideal product comes in. Right. So what does this ideal product need to offer? So this ideal product should offer speed. It should offer simplicity. It should also offer the ability for capacity for high throughput, right? Much like the crude extraction. So it should also offer purified and high quality nucleic acids suitable for all applications. It should have high yields, improving the efficiency of the workflows. It should also give you cost savings, right? Both in terms of labor, time, and consumables. So it should be compatible with all sample types. It adapts well with all sample types. It should have little to no PCI inhibitors. So this will ensure accuracy in your downstream molecular biology applications. Right. It should also be easily quantifiable via UV. So this will help improve your quality control processes. Right. It should also be automation friendly. So when you want to scale up, you have a lot of samples coming in. You can easily scale it up without changing the chemistry for high throughput capabilities. Right. It also should be more environmentally friendly than traditional purification. So it should reduce a lot of plastic and liquid waste generated. So this approach really eliminates the compromises of both crude and traditional purification methods, providing an efficient, high quality and cost effective solution. Next slide, please. Thank you. So all these led to the development of our SpeedX Lightning product. So this is an advanced magnetic bead-based nucleic acid purification technology that combines high speed, simplicity, and efficiency while maintaining exceptional DNA purity. Right, so this innovative approach really bridges the gap between crude DNA extraction and traditional purification methods. So it offers an ideal balance of efficiency, speed, and quality. Right, so unlike traditional magnetic bead-based methods that maybe some of you might be familiar with, right? So these traditional bead-based methods, they usually take more than 30 minutes, right? But SpeedX Lightning completes purification in just 5 to 10 minutes. And the process is streamlined into a 3-step protocol, saving you time and money. As it's also a magnetic bead-based chemistry, it will work with any open platform automation, right? Making it very automation friendly. It's also also very environmentally friendly as it uses less hazardous reagents and generates less waste, as we will see in the coming slides. Next slide, please. Thank you. Right, so in nucleic acid purification, the complexity of the workflow impacts the efficiency, the speed, and the overall usability. So let's compare different purification workflows here to understand how SpeedX Lightning optimizes the process. At the top row here, you can see this is your classic magnetic beads or spin column workflow. Right? So usually it requires 5 or more steps and takes about 20 to 45 minutes. And it usually involves multiple binding and washing steps, making it pretty labor intensive and time consuming, right? With all these multiple steps, right? So if you look at the middle row here, this is our current SpeedX workflow. So this is not the SpeedX Lightning. We actually have a traditional SpeedX workflow that we also offer. So this traditional SpeedX workflow, we streamlined it to 5 steps, right? And it takes about 40 minutes for purification. Again, it requires multiple binding and washing steps, but it also offers some improvement over the previous methods. So it's already a little bit faster than other magnetic beads workflow, but there's still room for improvement, I would say. So that's why we came up with SpeedX Lightning. This really revolutionizes the process by introducing a high-speed 3-step workflow, which will be completed in as quickly as 5 minutes. Right, so how do you achieve this? So again, the process is simplified. If you look at the bottom row there, the process is simplified to 3 steps. It's essentially your binding step, your wash step, and your elution step. So just 3 steps. So we eliminate a lot of the redundant washing steps marked in red here without compromising purity. Okay, so it also, one of the unique selling points, it also retains this water wash step. This is unique to our SpeedX line of products. Right, this water wash step, instead of using ethanol, we actually use water. So this skips the time-consuming ethanol drying step. Right, and it also ensures that no PCR inhibition from the ethanol in the final eluate. So let's say if you don't dry it enough, you will retain some of the ethanol, which will affect your downstream applications. Next slide, please. Thank you. Right, and just here we have a side-by-side visual comparison of SpeedX Lightning's protocol and against our traditional SpeedX, and then you can see why this matters. Right, the time needed for each step in a workflow has been drastically reduced, shortening the processing time. Right, this is perfect for labs with high throughput, where you need to keep up with a high volume of samples coming in with limited time in a working day. Right, so this optimized workflow makes SpeedX Lightning one of the fastest and most efficient nucleic acid purification solutions available today. Next slide, please. Thank you. Right, so looking at some of the value propositions SpeedX Lightning brings to customers to meet their requirements. Okay, so firstly, it will be simplicity. Right, protocol steps are reduced from the standard 5 to 3 steps. Right, this will save you time as much as up to 9 times when you compare to other kits. Right. Not to mention when you have fewer steps from 5 to 3 steps, you actually lower your chance of user errors during handling. Right, when you're working with a lot of samples, I think a lot of researchers in the labs will understand when you're working with a lot of samples and you have a lot of your manual protocol is very complex. You tend to make mistakes. Right, nobody's perfect. Right, so what we want to do is try to minimize that error. Right, making the manual protocol as simple as possible, so people make less mistakes. So all these will save you time and also save you money, of course. Right. So a higher sample throughput per day. Right. So your manual and your automated preps will be faster and simpler. It will also use less consumables, saving you time and money. Right. So we do all this, but we also make sure the purity, the quantity, and the yield of the nucleic acids will still be very high. So this will give you reliability for critical downstream applications and diagnostic sensitivity. Right. So on the right here, we have high flexibility. So high flexibility, we do this by using less reagents. We use less bottles and packaging material. It also weighs less. So why is this important? It saves a lot on shipping costs and also on storage space. Right, so with SpeedX Lightning, we save up to 80% less shipping costs and storage space. We also produce SpeedX Lightning also produces less hazardous waste. So why is this important? Because it will have less shipping or regulatory problems. Right, up to 70% less, and it saves consumables and shipping and storage space as well. Right, next, thank you. Right, so together with SpeedX Lightning as a new product, we also put a lot of thought into how the kit formatting can help potential users get started. Right, so we try to make it as easy as possible. So we developed something called the SpeedX Lightning Starter Kit. Right, so this starter kit process, it consists of 3 simple steps, and with that we work closely together with customers to ensure a tailor-made purification solution for every customer's specific needs. Right, so to briefly explain how this kit works, right? Each kit contains the core kit reagents, so it's either A or B. So we are looking at the left column right now. The core kit reagents A or B. Right, we have 6 different lysis buffer for testing compatibility. We also include other essential reagents for the chemistry, which includes the W capture beads and protease K that's needed for your purification. Right. So when you get your starter kit, first you need to run a few tests with your specific sample type. So by running these tests, you can determine the best lysis buffer to be used with your specific sample type, and whether the core kit A or core kit B is optimal choice. So essentially what you're doing is you're doing like a screening process for your sample type. So you're trying to see which reagents works best for your sample type, right? Once you've identified the best configuration for your specific sample type, you're ready for the next step. So looking at the second column here, now that you have optimized your protocol using our starter kit, right? You know which one works the best. So you select the specific components for your purification solution, right? And these will be created as a customized kit for you, right? So you after doing all this, you also include your optional reagents like your W capture beads, your protease K, your RNAs, right? So once you're done, you have this on the third column here. You will receive your customized SpeedX Lightning solution. So again, you only have to do this once for your sample type. So once you've done it, you know what's the best way to do it. In the future, when you're returning, your returning users can keep ordering the same customized solution for future runs to really simplify the ordering process. Okay, so next slide. please. Thank you. So why is this important? Right, so we try to offer this flexible format because it greatly reduces the number of our news bottles sent, which in turn reduces your shipping cost and your storage space needed. And we also save on the plastic and the chemical waste produced. So you throw away less things and you also produce less waste. So if you compare it with our traditional SpeedX kits, you can see in the pictures here, traditionally our SpeedX livestock complete kit, it comes with 12 bottles, 12 tubes and bottles, right? With our SpeedX Lightning core kit, we have reduced it from 12 to 4. So this greatly reduces the packaging material and also the amount of reagents that you will not use to a minimum. Right, next slide, please. Thank you. Right, so of course, when you're considering switching to SpeedX Lightning, the first question we will ask, is it comparable to our current methods, right, or other methods in the market, right? So in this section, we'll compare SpeedX Lightning with competitor purification kits, focusing on DNA purity and concentration. So SpeedX Lightning stands out due to its ability to deliver high-quality nucleic acids with superior purity, concentration, and integrity. So these are key factors for successful downstream applications like PCR and sequencing work. All right, so the table that you see here provides a detailed comparison of different purification methods. This includes our SpeedX Lightning, of course, our traditional SpeedX as well, and several other competitor kits. So we evaluated each method based on a few criteria. So the first will be our 260-280 ratio, using a nano drop. So this indicates protein contamination. So the ideal values here are between, I would say, 1.8 to 2 for pure DNA. All right, so we also have our 260-230 ratio for nano drop as well. So this indicates your salt or organic contamination. The ideal range here will be about 1.8 to 2.2. We also look at the concentration via qubit and nano drop as well. Right, so as you can see here, highlighted in the font in red, right, SpeedX Lightning achieves a strong 260-280 ratio of 1.83. This is comparable or better than competitors, ensuring minimal protein contamination. So the 260-230 ratio of 1.53 is also higher and comparable to many competitors, indicating an improved removal of contaminants. SpeedX Lightning also delivers the higher DNA concentration with 106 nanograms per microliter for the nano drop and 61 nanograms per microliter for qubit, outperforming all the other methods. And if you look at the gel electrophoresis images on the right here, when we're working with corn seed samples, you can see that SpeedX Lightning DNA bands are clear and intense, right, indicating high integrity and purity with high molecular weight DNA compared to other competitor kits. So if you look at other competitor kits, they show big bands or in some cases no bands at all. Next slide, please. Thank you. Okay, so in this slide, we will compare SpeedX Lightning with competitor purification kits in terms of the efficiency and sustainability. Right? So we focus a lot on the number of protocol steps, the protocol time per sample, plastic waste per sample, and the liquid waste per sample. Right? So efficiency in DNA extraction is crucial, not only for reducing labor and time, but also for minimizing environmental impact by cutting down on plastic and liquid waste. So let's analyze how SpeedX Lightning outperforms the competitors in these key areas. Right, so for protocol steps, SpeedX Lightning requires only 3 steps, the least among all the kits. So most competitors require at least 5 to 8 steps, making the workflows more complex and time-consuming. Right. We also have protocol time. SpeedX Lightning completes DNA purification in just 5 minutes. Right. This is significantly faster than competitors. Other kits take anywhere from 20 to 45 minutes per sample. And for plastic waste, SpeedX Lightning produces only 2 grams of plastic waste per sample, making it the most environmentally friendly option. Right, competitor kits generate between 3 to 19 grams of plastic waste, right? So some of them produce nearly 10 times more waste than SpeedX Lightning. And of course, we look at liquid waste as well, because this is important when you are disposing of your hazardous reagents, right? So SpeedX Lightning produces just 1.8 mils of liquid waste, the lowest in comparison, right? Other competitor kits generates between 2 to 5 mils, increasing the hazardous waste disposal requirements and cost. So you can see by switching to SpeedX Lightning, labs can significantly improve their workflow efficiency while also reducing their environmental footprint. Next slide, please. Thank you. Right. So again, some of case studies that we have. So we in LGC, we pride ourselves on ensuring that we partner with customers, right, to help and be involved in their journey to get started with SpeedX Lightning, right, through the whole optimization phase. So we have knowledgeable teams in place at each step of the process to help you navigate and optimize where it's needed. Right. So one such service we provide is to actually send your samples to our R&D team in Berlin for a pilot extraction study. Right. So this is where we work together with you to optimize your workflow with SpeedX Lightning, specifically to your sample type. Right. So what you see on the slide here is actually exactly what we have done with a few customers here in APAC. Right, they have sent their samples here to us in Berlin. Right, where we work together with them to come up with an optimized protocol for their specific sample type, and it has produced great results. Next slide, please. Thank you. Okay, so again, pairing it together with our other offerings. So if you remember that our workflow solutions are high throughput instruments, right? You can see in this example I have on the slide here, we have worked with fairly challenging bittergourd and papaya seeds that is commonly found in our region. And we utilize SpeedX Lightning with our high throughput instrument called the IntelliCube, right? So the IntelliCube is a first fully automated medium to high throughput instrument featuring seamless integration of liquid handling, thermocycling, detection, and data analysis. Right. So instead of your traditional 96 well plate or your 384 or 1536 plates, right, the IntelliCube uses an innovative array tape technology that saves a lot of time and reagents and plastic costs. Right. So you see on the left 2 pictures there. Data analysis was also done on our IntelliScore software, where you can examine the endpoint SNP genotyping clusters here on the left. And also using that software, we can also generate a report to show you the results. Right. So together with our IntelliCube and SpeedX Lightning, labs can produce up to 24,000 data points per day. So that brings you to about anywhere from 1 to 6 million data points per year. So you can see this is really truly a high throughput workflow here, right? So pairing it with our product offerings, we can really increase the efficiency in your workflows. Next slide, please. Thank you. Right, so that's all I have for you today. I'm right on the mark here at 30 minutes. So thank you all for your time and engagement today. I hope this session has given you some valuable insights on how SpeedX Lightning can enhance your DNA extraction workflows. And if you have any questions, I'll be happy to discuss them further. Otherwise, I also strongly encourage you to explore this technology firsthand, right? To really experience the benefits and advantages that it brings to labs. Right. And of course, if you have any questions after the session, my email is on the slide. I don't know if it's shown now. Yeah. So, but if you have my email, you can always drop me an email if you have any questions, or if you want to try out the technology, please feel free to reach out to me via email. And again, if you have any questions, please feel free to come forward and have a discussion. I'll be happy to answer any questions. Thank you so much.
Thank you very much, Brady. Especially for delivering the advanced technology for yielding high quality and quantity DNA as the vital starting material for molecular breeding, particularly for horticultural crops where many of the tissues are challenging tissues with high phenols, polysaccharides, and so on. So purity, the issue can be solved, I think, with this technology. To mention the molecular breeding, I think we'll be happy to explore the breeding program and associated technologies to address the complexity nature of horticultural crops mentioned by Prof. Dada earlier. And for that, we'll have Dusty with us. And to introduce you briefly, who is she? I love to call you Dusty. Awesome. And here's the brief CV of Dusty. Moment, please. Yeah. Okay. So Dusty was graduated from the University of East London in applied biology and plant molecular biology. She has a long experience as a researcher in many institutions. I believe from Biochem for 12 years and for the University of York for 5 years, and as a plant genetics specialist in the LGC diagnostic and genomics from 2012 until present. So she will share with us to explore the molecular breeding programs using genetic selections and associated technologies. The time is yours.
Thank you so much, and I am very, very excited to be here. So, as you said, I revised, and actually the dates were wrong. I actually started my degree in 1989 to 1994, and it's around 36 years now. So it's been a very, very long. So can you go to the second slide? Just go straight to the second slide. Okay. And the next one?
Yang besok udah yang itu yang delay.
Perfect. Okay. So as I was saying, I've been doing this a very, very long time now. So, and I've seen some amazing advancements in genomic technologies. And as Professor Dada of technologies that have been applied. And if we can I have a pointer, or I'll just use this pointer actually. Oh no, I can't use this pointer. So when I started in 1989, I was using SSR markers. I was developing them. I've used rapids, and rapids are random. It's in the name. So so for me, they have a function, and that now still SSRs still have a function at the minute, but AFLPs and RFLPs are technologies which were useful, and along with conventional breeding, they showed us the path to take with molecular marker development and the strategies to use in breeding. So we then moved on to a PCR-based options, and this was a landmark change, which is what I call it. And that, chemistries such as Cas genotyping and Tapman assays were introducing the capability to apply effective marker-assisted selection breeding. But, these were in the late 90s, early 2000s that these were developed. And then in the early 2000s, we also saw a key next generation sequencing. So what these did was that they enabled breeders to use higher resolution markers, which in turn enabled them to understand the genomes that they were working with. So the solid state chips and arrays, which were developed in the late 2000s, finally gave breeders a route to look at effective genome-wide association studies. And it was great. They achieved a lot, but in reality, they were very expensive to develop. There was a lot of ascertainment bias in the SNP chips. Maybe I should test you and see if anybody knows what ascertainment bias is. No, I won't do that. So ascertainment bias is actually where you only can use a proportion of the markers effectively. So there is a bias towards the markers that are not useful to you. So when you're looking at a solid chip, that could mean the loss of maybe 20% of data that's generated. And generally, the arrays were developed around varieties which were not necessarily relevant to breeders around the world. But, they were effective and, it showed us a different direction. Then back in 2017, the concept of genomic selection was discussed and first implemented. So for that, we needed technologies that could enable that higher throughput application of molecular markers. And the beauty of genomic selection, it basically takes the guesswork out of the selection process that you are trying to carry out. So you are using sequence-based markers along with phenotyping to accurately select the plants to take forward in the next generation. And ultimately, the impact of this was we were increasing the genetic gains in the varieties that we were developing, and we were doing it quicker. So that's still fine, and things are moving forward. But I think we're in a different moment now. We're moving into a new era, and as Professor Dada correctly said, that we are moving into an era where the artificial intelligence is being used, machine learning is being applied to technologies, and genotyping technologies are continued to be refined, and that's what we're about. That's what we do as a company. We enable the customers to use the latest technologies cost efficiently, effectively, and we work with the scales that are applicable to you as breeders. And maybe in the future, you guys are going to, use these technologies every single day. And in fact, it was funny, we were talking in the car on the way here. We've done a lot of development in the past 30, 40 years. Now it's up to you. You guys have the responsibility of carrying this torch to enable us to adapt to climate change much, much quicker, because it's something that's going to happen and it's unavoidable, and you have the capabilities to make that change yourselves. So anyway, before I get on my soapbox of talking about saving the world, so as I was saying, we have developing genotyping technologies that are enabling an increase in the simplicity of how you're applying NGS technologies. We are developing the formats which are going to be useful to you to use in your own laboratories. But I won't get too excited, and I'll talk about that in a moment. Next slide. So when thinking about the implementation of target, genotyping sequencing technologies. Obviously, the most ideal approach is to have whole genome sequence information. That means you understand completely what that genome is made of. You understand the structural variation. And you have the full story of what is happening in your individual species, in your individual varieties. But of course, at the moment, whole genome sequencing is very, very expensive. So what's the next best approach? There are options to do RNA-seq expression studies, exome capture. There's standard genotyping by sequencing, which is very applicable to people who do not have a reference genome. So something like GBS is still incredibly relevant at the moment. So it would save you money on not having to develop a whole reference genome. But for me, one of the most effective developments in reduced representation sequencing is actually targeted genotyping by sequencing. And so TGBS, as I'll call it, because I can't be bothered to say targeted genotyping by sequencing all the time. So TGBS does currently provide the flexibility to apply a broad range of applications, including genomic selections, all the way through to quality control and parentage. So I wanted to introduce you, to the cost effectiveness of TGBS. So when we are, sorry, next slide. I forgot I'm doing it on here and I feel like you're doing it automatically over there. So it's really important to understand the populations you are trying to screen, and there is an inference of population structure and the appropriate genomic technologies to apply. And professors that have said it perfectly before as well. So when you're looking at phylogeny, when you're looking at relationships between different lines, it is possible to use a very low marker set, and you can use that very, very effectively. So what we regard that to be, there is divergence. The divergence is limited. So you can use smaller numbers of markers. But then as you move through things where recombination is limited and you're taking into account things like linkage disequilibrium, and you are trying to identify the genes which are controlling the phenotypes and phenotypes of your desirable traits. So it's important to understand that this is a medium resolution capability. And it enables you to identify functional mechanisms in QTL or pedigree mapping. So when we're looking at association mapping, and it is in this range of high density markers and where the linkage disequilibrium is limited, we need to apply much higher resolution screening for your populations and your lines. But this does give you a better understanding of what is happening in your genome with greater accuracy. And obviously, this is an example of the strategies that can be used when thinking about genomic selection. So genomic selection is ideally suited to where you can accurately associate your markers and the phenotype. So genomic selection, we rely on genomic estimated breeding values. And that's established through training populations. Next slide. So let me talk a bit about variety development. In fact, you are the experts, and I'm not the expert on this at all. But teaching the next generation, it's important for them to understand the basics of how you take a wild variety all the way through to a commercial crop variety. So when we look at the considerations, you know, you have to understand the pollination method. Is it cross-pollinated or self-pollinated? So what we see generally in wild varieties, there is a high degree of genetic variation and very low trait uniformity. And as you know, farmers and smallholder farmers may go through a strategy of saving seed. That is a selection process in itself. So what happens then? You have the early varieties being named as land races. So the degree of genetic variation is reducing and the trait uniformity is increasing. But ultimately, when we are trying to feed the world, we want, you know, when we want all the fruits, that delicious fruits that we want to eat, we need to ensure that the farmers can have that trait uniformity to ensure that they can grow enough of the crops for their smallholder farmers. Or if we look at larger scale, that uniformity is for the plantations where the whole world relies on those crops. So this is about, you know, the consideration you guys need to think about is how you're going to approach the variety development. Next slide. So we've heard about marker-assisted selection, and I hope you've all studied it. So what I wanted to do for you here today was to give you a comparison between marker-assisted selection and genomic selection. And then, with marker-assisted selection, you can do some pre-breeding, which is fine, and that could be almost considered a training phase of your markers. But we are looking at specific QTLs and a lower density set of markers. So we're still trying to associate that QTL with that phenotype specifically. But when we look at genomic selection, this relies on a training population. And back in 2015, 2017, the idea of a training population size was in the range of 200 to 300 lines to be tested with a marker density of around 2,000 to 3,000 markers. But things are changing. You know, there is evidence now that is showing that to truly understand the very, very complex trait, particularly things like Professor Dada was talking about post-harvest shelf life.
No, you can continue.
Okay. So looking at increasing shelf life, you showed the genes that were involved, the complexity of it. What is happening now is that we are seeing, by increasing the that training population size and increasing the marker densities that are being used on that population, it is making the selection and identification of the plants that you actually have all the traits, all the complex traits you need more accurately. So having a detailed understanding of the contribution of those SNPs to very, very complex trait is absolutely feasible now. So that's the training phase. But the amazing thing about genomic selection is that once you've identified in the training population the SNPs which are associated with your complex trait, when it comes to actually your breeding programs, you don't necessarily have to screen and genotype every single population. So that means you can base your selection on markers which have a statistical high value and provide you with a lot more accuracy, which ultimately means you are saving time. You are saving field costs. You're saving, you can develop varieties in half the time potentially. And then, you know, and we do not have time. You guys, you got to do something. You got to embrace these new approaches and take them forward. So actually, when the breeding phase, you actually looking at genomic selection models, and you're basing your selection on those estimated breeding values rather than a guesswork of low resolution markers and what can sometimes be a subjective selection process. Next slide. So when we are considering genomic selection, and actually I feel like we had a very similar talk, Professor Dado, because I'm going to talk about the same things. So the key criteria, in my opinion, when looking at sequence-based technologies are the consideration of the genome size and the complexity. Banana, of course, we know is incredibly complex, but it's feasible now because, you know, rather than by using high density markers, you're enabling that accurate association. The availability of reference genome for a TGBS approach is absolutely critical. The other important aspect is the heritability of those traits. So higher prediction accuracies in the training data are better represented when there is a strong relationship in the varieties that you are testing in your training population. And higher marker densities are also critical when predicting very distant crosses. So when you are trying to integrate a trait from a wild variety or a landrace into your beautiful papaya variety, then you need to increase the numbers of markers because you are taking two very distant and very variable genomes into one plant. So ultimately one new variety. So we, you know, these are the considerations that really do need to be taken into account. Next slide, please. So, you know, genomic selection, as I said before, has been, you know, effectively applied since about 2015. So we do actually have data, and there's a huge amount of publications that show the impact of genomic selection on breeding programs. We've seen that selection based on GEBVs means that you are taking out the subjective selection of traits and plants. You are able to achieve higher rates of genetic gain. So when we started out doing genomic selection, the global overall global genetic gain for all varieties was between 0.4 and 0.6%. The expectation is if we need to produce enough food for the world in the next 20, 30 years, that needs to increase tenfold. And the reality is that we are doing it. And in fact, it's better than 10%, 10 times more. So it's begun to be an established process, and it's fantastic. You know, we are pushing the boundaries forward. We are making these selections in a reduced time. There's reduced cycling time. There's an increase in the selection intensity, and we can understand the extremely complex traits better. One of the key factors for me when understanding genomic selection with a very good reference genome is that you can understand structural variation. So structural variation is understanding actually what is in your genome and the relationship of those genes with those, for those particular traits. in a physical manner, so you can get a true association and understanding of structural variation. And of course, the beauty of genomic selection is that you can incorporate historical data. So all the work that has been done over the years still can be of value and can add to your genomic selection programs. Next slide. So what's needed next? What do you guys need to do next? Not me. This is what you got to do. Okay. I'm giving you a list of challenges for the next 15 years. We need to look at how to change the capabilities and how are they going to impact the breeding programs. We need to save time. We need to make it cost efficient. So when thinking about genomic selection, one of the key factors which is becoming a bottleneck is computing power. The amount of data that's generated using sequence-based technologies is huge. Do you have enough computing power to actually run your sequence analysis on your computer? How many days is it going to take? Or do you need those answers in hours? So these are considerations for the future that really need to think about the capacity. We need to increase the accuracy. Obviously, there are technologies out there which give good quality data, but when we look at the increases in the accuracy of that data, as little as 10% increase in accuracy in the metrics that are developed through data generated through next generation sequencing can have an impact. So that 10% means that you can potentially take fewer plants forward. You can have greater confidence in the data that's generated. And it can really make a difference in how long it's going to take for you to select your most accurate line to take forward. We need to look at the scale, how much testing is needed. We need to have simplistic workflows that anybody can use. And of course, we need to think about future applications such as AI. So by utilizing artificial intelligence to predict breeding relevant information associated with complex traits, we will begin to understand environmental and genetic relationships. Artificial intelligence will also enable researchers to conduct complex studies much, much easier. There'll be less effort, while automation will significantly enhance the experimental accuracy and efficiency. And in addition, what we're seeing in plant breeding, not in only the large companies, we are seeing rapid and changes and breakthroughs in the era of data explosion. And this is a signal for the beginning of smart breeding, a smart breeding revolution. Next slide. So I can't believe, I'll hurry up. I realise I've been talking nearly half an hour now, but I'm trying to educate you guys. So what's next? What will these impacts actually have on the future? So it will have the impact of the capability of opening genomic selection to smaller breeders, smaller breeding programs, what I'm trying to say. It's going to increase sexual selection accuracy. There will be less number of plants to take forward in each cycle, leading a reduction of costs in field costs without losing the power and without losing that power of selection. We need to look at the capabilities. We need to be, we are developing chemistries which can have a great impact on individual breeding programs capabilities. this is where epigenetics is really, really going to kick in. This is going to enable us to understand that missing jigsaw piece that has existed in my mind for such a long time. And I want to find a solution to that. Next slide. So if we look at who we are as LGC Technologies, we are a company that has always looked forward. We are a company that is always trying to help find solution for breeders. And hence, that's why we have, and proudly we can say we are a company that's looking at science for a safer world, because we are looking to the future. So in this matrix, what we have is the capabilities that we have in-house, and there are options to run it in-house, and then we are developing kit formats which you can run in your own laboratory. Brady has beautifully talked about all of our extraction chemistry, so I won't bother talking about that. So when we're looking at genome exploration and SNP discovery, we have capabilities such as whole genome sequencing. We have CaptureSeq, which is a service-based option, which is where there is incredibly high density of markers, and that can be applied to a range of different sample numbers as well. We've got FlexSeq, which is our mid to high density capability. And currently that's offered as a service. And this is a really good option if you want to apply NGS technologies without the investment of very expensive sequencers. So basically you would send the samples to us, we would do the panel design, we'd do the sequencing, and you would get the data back. But the new baby, or my baby, should I say, is AMP-Seq. So for me, AMP-Seq is a game changer. So as I was saying before, we're at this crossroads, we're at this new era of capabilities that are being made available to the breeders, and it is a workflow solution. And ideally, with AMP-Seq, the numbers of markers can be anything from 50 to 5,000. So this is useful for when you're looking at, medium resolution, but it can also be used for looking at imputation. If you have identified a set of markers that from a large set of markers, a subset that can be used effectively. So that is what we call imputation and haplotyping. So AMP-Seq is a kit which you can run in-house. It's a very effective workflow, and I'll talk about it in a bit. And then, of course, we have the high throughput instrumentation where you're looking at million plus samples. We actually worked with the UK government during COVID, and our instrumentation was applied as a solution for the government to produce effective screening for COVID. I always forget about that, actually, but we did do that. Maybe it's a nightmare. So next slide, please. So this is it. This is the game changer. This is where everything changes. So AMP-Seq is simple, it's efficient, and it's robust. So, where am I? So this is a proprietary chemistry that we've developed in-house, and I'm really proud of our R&D team that have produced this chemistry, but it was made with a key industrial partner. Our R&D team developed the panel design software, the chemistry, and also the analysis software. So this is a totally organic development of chemistry and workflow. As I said before, it's effective from tens of markers to 5,000 markers. And the difference also with AMP-Seq is the fact that it can be used with both crude and purified DNA. But when coming to the crude DNA, there would need to be some, we would need to carry out validation and optimization as well. The benefits are just incredible. So we are looking at reducing the sample to sequence turnaround time by 50%. And anybody who's run any kind of NGS will understand that rather than doing things in 3 days, you want it done in the same day. And we can do it now. So we're reducing the total workflow costs. So because we're saving on consumables, labor, and sequence accuracy and sequencing. So as Brady said, we are a company that looks to the future, and we are going to future-proof the technology and give that capability of global distribution and secure supply. Next slide. So what is AMP-Seq? AMP-Seq is an in-house, so it's a kit, targeted genotype by sequencing workflow. As I said, I've talked about the numbers of markers. So what's included in our AMP-Seq workflow includes the panel, includes the core reagent system, it includes the index plates, callers, index plates, and it includes our biosearch caller pipeline, which if you decide to convert to AMP-Seq, it is something that we would give for free. I'm looking at you just to make sure I'm correct on that. Oh, she's the boss. So, and it's very effective for looking at genomic selection, parentage tested. People use it in plant and animal breeding as well. So the service that we provide is that we do the oligo design. We can do the design for you, and you can have that panel very cost effectively, and you can optimize it yourself if you wanted to. You have the option for us to optimize that panel for you if you want us to. So we call that a panel verification stage. You would send us your samples and we would find the optimal probe concentration and DNA input for you to apply in your testing process. It's automation compatible. So for the large scale breeders, this is incredibly important because there are, as I said before, there are some of our customers who are running 2 to 3 million samples a year. The analysis pipeline, which I've already spoken about, and we have customization options as well. Next slide. So what is it? It is as simple as this. So in the box, what you get is that you get your assay reagent, you get your index plates, and you get your master mix. All what you need to do is make your reaction mixture of your assay reagent, your master mix. You dispense your DNA onto the provided index plate, which is part of the kit. And the amount of DNA you need to add is 1 microliter. So at a concentration of potentially about 5 nanograms. So very, very low DNA input, or it could be crude. So when you look at other technologies or our competitors, the amount of DNA that you need to input into the reaction is fairly high. So you've done your master mix, you've put your samples in a 384 format, and then you put it on a PCR machine. And that PCR cycling is around 50 minutes. You then you pull those samples into a single tube. You carry out a magnetic base cleanup, and then you put it on your sequencer. Next slide. So this is it. This is how long it actually takes. So it's a very, very streamlined library prep. So if you're doing it 384 by hand, which I don't know, I hate it myself because it's so fiddly. But of course, this is all automation compatible. It takes about 25 minutes to actually set up your reaction plate. The PCR cycling, as I said before, is 50 minutes. And the pull and cleanup stages is about 45 minutes. And then you put it straight on the sequencer. So what we have done here is effectively we have developed a NGS library prep in the same time as you would do a PCR endpoint, single PCR. So the cost efficiency that is introduced using this system means that rather than getting a single allele in one well, you're getting potentially 5,000. Just let that sink in, because that's the magic. You're getting 5,000 data points for the same time frame as you would get from a single endpoint PCR reaction. So, next slide. So when we talk about the panel design, and for me, this is absolutely critical. If you have a good panel design, it will mean that you're going to get better quality data, and it will increase the longevity of the effectiveness of that panel. So again, we provide flexibility in this area as well. There are options for single end reads or paired end reads of 150, which means that you are introducing efficiency into the cost of sequencing. So with a single end 100 base pair, a sequencing length means that on a single flow cell, you're going to be able to put more samples. You can choose the distance of the targets to the according to your sequencing needs primarily. We provide our customers with options of designating their priorities. So you can have priority 1 markers, which are absolute must-have. So these tend to be QTL or disease-resistant markers, and priority 2s, which, you know, the design process, if we were to drop any of the markers, it's likely to be either priority 2 or priority 3. But in all honesty, the quality of data we're seeing, we're not having to drop that many markers. The capabilities of what we can do, we can design SNPs, multiple nucleotide polymorphisms, indels, and it could be used for CRISPR screening. So when you produce your CRISPR edited plants, you can screen for the presence of your site-directed mutagenesis, your CRISPR edit, as well as assessing any off-target insertions that may have occurred. So rather than doing a 2-stage process, you can actually screen and identify the plants where you have a single copy of your variation with no off-target insertions. Next slide. Thank you. So the AMP-seq workflow is design-centered approach because, as I said, having a really good panel design makes the world a difference. We ask for the flanking regions for your specific targets. Then we design all the possible flanking potential probes in the flanking regions. Then we go through a filtration step and ranking of the targets, and it's these markers then we take forward for the panel design. So through this process, we do produce incredible results. So next slide. I'll make sure he wakes up in a minute. Okay. So these are kind of the typical metrics that we're getting overall when we've looked at bovine, we've looked at canola, maize, sorghum, soy, strawberry, and wheat. So I apologize, there's not that many fruits in here, but hopefully we will change that in the future. And this is very, very new product. Yeah, that's the most important, because I love strawberries. So what we are actually seeing, we're seeing call rate metrics between 90 to 99.5% at a mean depth of sequencing of around 200. We're getting between 80 for the more complex species and over 90% of coverage uniformity. We're getting greater than 98% concordance with alternative technologies such as CASP genotyping, even SSRs. We're getting concordance with SSRs. And as I said, so they're the species that we've worked on. So when we look at the robustness, so we're comparing both purified and crude extracts. If you compare the data that's been generating, we're getting 98.26% concordance between purified and crude DNA. Looking at the mapped reads, the reads on target, targets calls, sequencing depth and uniformity, there really isn't much difference when we were screening the samples, the soy samples with this 1,000 target panel. And we, of course, we have the analysis pipeline, which is, oh, sorry, next slide. We have the BioSearch crawler, which is a proprietary, it's not proprietary actually, it's a software where we have taken publicly available tools and streamlined to enable fast genotype calling. There's options to configure the call parameters. You can look at optimizing your own bioinformatics workflow. We will help you adapt the BioSearch crawler to that. It carries out downsampling, trimming, alignments QC, and the results are generally provided as a VCF file, or you can have it as a simple genotype call. But the great thing is that rather than getting your single genotype call, you will actually get the surrounding sequence information, which could be absolutely essential when understanding subtle minor allele frequencies which are contributing to your individual traits. And of course, as I said, it will provide you with de novo SNP discovery as well. So overall, when we're looking at, what AMP-Seq does and what we do, we help with the development. We work with the customer we have all the options. So we have the option of just doing the design for you. We have the option of doing that as a pilot panel verification, and we produce the kits for you. You can then have the option of testing these kits in your house to see the concordance between our lab and your lab. And we help with pre-production testing as well. But ultimately, we're looking at implementation and production scale screening. Next slide. So for me, AMP-Seq is definitely a game-changing technology. So that's when I was talking about we are moving into another era, and I truly believe AMP-Seq will change breeding programs. It's a effective, efficient, simple workflow. We've got incredible accuracy with data metrics. The data speaks for itself. It's just easy. You can get extremely complex trait evaluation. We've looked at the fact that the labor costs will be reduced. It's environmentally friendly because you're not having to have additional plates. For me, when breeders think about it, it may enable to make your selections earlier in your breeding programs, in turn reducing field costs and enable scalability. So, okay, I've got 10 minutes to talk about CASP. So next slide. So CASP genotyping is an alternative. So if NGS seems daunting, the computing power, the understanding, learning bioinformatics, may be difficult. So for me, CASP as an introduction to applying high throughput efficient technologies is the perfect answer. So what we have in our kit is we have 3 components. We have your allele specific primers, which actually has a tail which contains the sequences to specific. It's a fret cassette, which is complementary to the fret cassette that's included in the master mix. And of course, you need your DNA template as well. So next slide. So how does CAS work? So in the first round of PCR, you get annealing of your allele-specific base to your target SNP. You then get elongation of that in your first round of PCR. And of course, you get your common reverse primer binding to the strand that's been denatured, and you get elongation of that. So you get a 2 copies of your strand. One has been targeted to the specific SNP, and the other one is the common reverse primer, which will be important in the next round of the PCR reaction. So next slide. So that first strand where the common primer was attached effectively, this will then elongate and make the complementary strand of that first allele, which was able to bind to your target SNP. Next slide. So in the third round of PCR, what happens is that once that strand has been produced, you get annealing of your the fret cassette. What's it? So the fluorophore on that oligo with that tail, the FAM labeled oligo, is actually complemented and is no longer quenched, which means that it will fluoresce. And that's what you detect in a CAS reaction. Next slide. So one of the great things about CASP is that it can differentiate different targets. It can differentiate SNPs, small indels, large indels, and single insertions or deletions as well. It's a very straightforward PCR thermal cycling. It's a 2-step PCR protocol rather than the typical 3-step protocol you get. Oh, sorry, next slide. I think you keep reading my mind and you're moving on with me, but obviously not. So it's a very straightforward PCR, as I was saying. It's a 2-step PCR protocol rather than the 3 traditional amplification stages that we have in a non-touchdown PCR. So typically CAS involves 10 cycles of touchdown and where the anneal temperature is reduced gradually. This improves the specificity of the reaction. And then we're followed with 26 cycles of annealing and extension that provides you with enough copies of your target sequence. And then ultimately we reduce that temperature down to less than 40 degrees to ensure we get efficient. I can't remember that bit. So it's something to do with fret cassette. Forget that bit. I can't remember it. So let's move on to the next slide. I've got 8 minutes left. So how does CAS work? How do you visualize your data? So rather than getting massive sequencing files, it's a very, very simple way of identifying which allele that you have amplified in your reaction. So on the y-axis, we have the homozygous that is only detected with the hex fluorophore. On the x-axis, we've got the FAM. Yeah, it's FAM. I can't see because his head's in the way. I'm joking. So on the x-axis, we have our homozygous allele, which is only detected using a FAM fluorophore. And using this simplistic cluster analysis approach, you can then also identify clearly, if you've got a really good CASP assay, all the heterozygotes. So it's a very, very simple approach. So quickly talking, sorry, next couple of slides. So this one, yeah. So an example where we have used all of our technologies is we carried out whole genome sequences on 9 indica genotypes, and this was done in collaboration with Nepalese breeders. And we were trying to target 2 horrible resistance genes. So we carried out whole genome sequencing. Next slide. And we were able to increase the coverage through this SNP discovery process. Initially, we had around 491 available CAS markers that were indica specific, not japonica, that had 100% identity. And after the whole genome sequencing, we were increased the coverage by 2,740 times. So this increased the density of the mapping and understanding of that genome. Next slide. So what's happened is that using, next slide. Sorry. Oh, okay. Sorry. Yeah, that's it. So what we've done, what we've been able to do with the whole genome sequencing, we were able to design nearly 5,000 CASP assays, which were then, which was successfully validated on a large number of lines, including japonica as well. So we were able to do, able to discriminate 178 genotypes with over 3,000 CASP assays. The success rate and validation was increased due to the fact that we understood the variation in the surrounding sequence, and we were able to incorporate that into the designs. We then carried out a TGBS option, but this was using an old technology called SeqSNP, which has now been replaced by our FlexSeq capability. Next slide. Nearly there. So what we were also able to do with the CASP assay designs, that we worked with the UK government, and we were able to carry out analysis for the UK government to identify true basmati rice varieties. And we were able to do that with only 327 CAS markers, revealing 255 polymorphic loci. And the other thing we did with this, that we were able to replace not all, but the majority of SSR markers with a PCR-based screening method. And all of this can be, if you want the links there, and maybe I'll be able to provide you with that later. Next slide. Yeah. So in summary, and I'm at 58 minutes, we are future-proofing the gains of genomic selection in breeding programs. We are able to produce better, faster, cheaper workflows to accelerate genomic selection in plant and animal genomics. We have the capability of developing robust panel designs and ensure longevity and the applicability of the targets, increasing accuracy. We've improved the NGS workflow, so it's the same time frame as endpoint genotyping or a single PCR reaction. It is enabling breeders to rethink their strategic decisions in their breeding program. And ultimately, AMP-Seek is taking genomic selection to the next level. Last slide. There you go. So thank you so much for listening to me. I hope you've learned something, you guys. Okay, you're going to do it? Promise? Okay. Perfect. Thank you so much, everyone.
Thank you very much, Dusty, for such inspirational talk. Yeah, especially that you mentioned that molecular technology has a speedy development, and also to explain variety of associated technology in the field, including the AMSEC as your baby. So now we are coming to the question and answer session. With that, I would like to invite again Professor Dada to the front. And please, for the participants, I know many of you have the question in mind, including those in the online. For those who are attending on site, please raise your hand. If you have any question, one question. And okay, we start with one first. Can anyone help with the mic? Marisa, please. Okay. Thank you. Please kindly mention your name and to who your question is addressed to.
Hello. Okay. My name is Wirio. Actually, I am from LPPOM MUI, laboratory. So actually, I am one of the stakeholders to develop extraction kit magnetic. Actually, we try this year. We try to develop extraction magnetic kit in-house in Indonesia. And actually, we already work and discuss with Saitiva and also Saitiva Singapore and Thermo Taiwan. And I'm happy to meet LGC, and also I'm not fully aware that LGC work with ScienceWork. If I know, maybe I will invite you for discussion. Because LPPOM MUI this year want to try to make in-house extraction magnetic for food metrics. Actually, food metrics and plant metrics is somewhat similar. And I'm aware that the CBIT-X Lightning also offer 6 type of bioanalysis, right? I think 3 of them is containing CTAP. Yes, containing CTAP, and 1 containing lithium buffer. One of my question is the first one: do you sell the magnetic bit only? This is the question. The second is how customizable the kit, the CBIT-X Lightning is. And also the last question is can we incorporate the typical lysis buffer, washing buffer of silica column reagent with the magnetic bit? Because one of my discussion with Saitiva Singapore, actually one of the approach is we want to try to develop the existing reagent. Actually, in Indonesia, a lot of them use silica column. This product is one of the biggest market in Indonesia. Magnetic is the lowest, I think, because they think it's far more expensive and automated, right? But we try to change that in the future. We try to change that by using manual DNA extraction kit for magnetic. So it far more cheaper and also can work with low quantity sample. So do you think that we can incorporate the typical lysis buffer from other company with your magnetic bit? Because I think it's somewhat similar method technique for silica and also magnetic bit. So
Right. Thank you so much for sharing. So, to answer your first question, I believe we are able to sell just the SpaceX Lightning beads, right? But again, but we cannot guarantee the efficacy because SpaceX Lightning was developed quite specifically for our reagents. So we always will recommend users to follow our reagent system, right? Because all this has been tested and we know it works, right? So you also mentioned that you've been using it with food samples. Yeah. So because SpaceX Lightning is a pretty new product, so we have done a lot of testing on plants, right? But not too much on food. But I'm still quite confident that it will work because we have done food sample our traditional SPX as well. So it has worked. So I'm quite confident it will also work. So what I would suggest is for you to maybe get in touch with Science Work, right? Maybe get a few starter kits to really get started to just give it a shot to try and look. We'll also be quite curious to see what the results are because we haven't really tested it too much on food samples as well. Right. So for I didn't catch the second question. What was the
third one?
The second one.
The second one.
The second question? Yeah.
How customizable the kit is?
How? Sorry.
Customizable.
Yeah. So basically, we made it very flexible. We are able to customize it according to your sample volume, right? And also the reagent configuration that you need for your particular sample type. Yeah. So I would say is we can work together with you to come up with something that suits your needs. Yeah. So the third question is whether other lysis buffers is compatible. So again, we developed it solely with because our SpeedX beads they are proprietary. Yeah, they're not they're different from competitor beads. So we cannot guarantee how well it will work if you pair it together with other lysis buffers. But of course, you are also welcome to try. Right, if you purchase our SpeedX Lightning beads, you want to try it with other lysis buffers. We can support. I wouldn't say we can fully support it, but we will support you. Right, we will try our best to see how we can make it work. But obviously, there's no guarantees because we did not do any tests and we develop it solely with our reagents in mind. Yeah. Hope that answers your question.
Okay, thank you.
You can feel free to drop me an email if you want to discuss further. We can take it to the next step. Yeah.
Okay. Yeah, sure.
Thank you very much. And maybe we can welcome some questions from the online participants. Can please the committee share something on the screen? Okay, for Prof. Darda. Okay, so I think the question is, what are the challenges and obstacles to durian breeding and donation of durian? And technology in general, there is still a lack of research for durian. So, Prof. Dara, I think this is on your field of expertise, please.
Thank you for the questions. So the first question was the challenge and obstacle durian breeding and biotechnology. It's what's up to now. There is still a lack of research. Yeah, as tropical perennial crops, our durian is actually in nature, it's a lots of genetic variability. So which one we will do the breeding for what's target of the breeding? I think for durians, maybe remove the odors, something like that, so everybody can eat durians without the smells. Yeah, but I don't know. But which genotype we will, because it's too many genotypes, too many says, too many varieties. if we are doing the wrong, choose the wrong starting points, that will be also will problem in the end of the breeding cycles. So yeah, the problem is too many. We have variability, too many variety. Maybe also the heterozygosity of the durians is also the problems. And then the next problem is the long juvenile periods of the durians. So if we start breeding today, maybe the result will get the fruits maybe in the next 15 years. So for the breeding of the tropical perennial crops, that's the big problem. We will give funding for the research for the long-term breeding. That's the problem. The second one is almost gene edited through genetic engineering. How can we predict and avoid the edited genes affecting other physiological process in the horticulture crops? Okay, if we are doing genetic engineering with the transgenesis or the old technology, as everything is random. So we don't know where is the genes integrates in the genome. So it's so random. And also there's some position effects. If the genes we are introduced to the genome, the new transgenes, and will affect the endogenous genes. So maybe effects for the already good traits and then silenced by the new genes is also the problems. Even with the targeted CRISPR systems, it's also off targets. It could be happen. So we don't know actually. We're just doing many transformation or many gene editing, and then selects the best ones for us. Selects the best traits, and then looking for the all good traits is not silencing. So other physiological process in the horticulture's crops. Yeah, I think many possibility could happen. The good things, the bad things, but back to the selections, doing a lot of transformation or genome editing, and then selects what the best for us, because other things is outside our capacity or capability to force the genes to go to the exact locations that we need. We cannot do as yeah, we are not God, so we are yeah. So the random thing and makes the selection. Thank you.
Thank you, Prof. Darda. Yeah, we have so many possibilities during the breeding process and the molecular breeding for to be precise. So do you have any more questions from the forum? Yes, please. Please mention your name and your question. If it works, yeah, you can use the mic behind you. Thank you.
Oke, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. My name is Sriwindyarti, but you can call me Windy. I am a bachelor student of agronomy and horticulture. So I'm here with my friends. We are the only bachelor student here, I think. So, as a Gen Z, I have a really good relationship with AI or artificial intelligence. And Dusty, oh, just Dusty. Okay, sorry. Dusty, you mentioned it before that the AI is possibly will be applicated in biotechnology also. And I want to know your perspective about this one. And do you have any prediction about AI application in the near future, maybe for PCR or molecular models, etc., etc.?
Yep.
I think that's very interesting, yeah, when we can use AI in biotechnology. Okay, maybe that's all for me. Thank you for all the insight that you gave me. And this is such a big honor for me actually to attend this event. Thank you so much.
Thank you very much for your very good question. So the reality is it is happening now. So there are smart breeding programs already occurring. So what is happening with AI and smart breeding strategies? They are using these huge databases to mine information without having to do all the bioinformatics yourself. It's using historical data. And it is already happening. There are companies who are developing AI breeding approaches. It's here. So if you Google AI breeding, you'll get a whole load of papers where people are using it already. And another place where it's being used effectively is in the genomic selection modeling. So when I was talking about using that high density set of markers with your high sample numbers, the amount of data that's generating is huge, but it's taking that data, looking into public databases, it's using global information to understand that genetic change much more accurately with less effort. It's here. We don't have to wait for it. And I'm so excited for you. Imagine what's going to happen in the next 30 years of your career. It's going to be brilliant.
Thank you, Dusty. And as you're explaining, I'm trying to Google AI, and there is a review paper about artificial intelligence in plant breeding. So you guys in Gen Z, you can see the papers and to see what kind of AI platform available for plant breeding in the future and make change with that. Okay. Yeah, it's there, really. Okay, we are running out of time, but I think we can come with some more questions still from the online. For Brady, okay, let me check how to identify if I make this method to the linkage mapping model to specific gene dimensions, beta-carotene metabolites compared to corn population. Yeah, I think it's Brady, do you get the question?
Is it lysine?
What is that? Lichen?
Lichen.
What is that?
That's the moss. It's like a plant on trees and lichen.
Moss. Okay.
Yeah.
Moss.
Yeah, but I need to know what is the resonance.
I think that's probably more to me than to
Yeah, I think the question is more related to Dusty or Professor Darter.
Maybe I can answer number 2, but number 1 might.
Or maybe please, because it's question for Mr. Han. Because you can address the relevant question for you, and then we would like to invite other speakers to respond to the other questions.
Right, sure.
For question one, I'm not an expert in breeding, so I will leave it to the experts, the other experts here. So for question two, so again, this really depends. The costing wise, it really depends on the sample volume that you have, right? So it will be it definitely will be a little bit more expensive than more traditional extraction methods like maybe CTAP, right? But what you're gaining in terms of to exchange in exchange for cost is simplicity, right? Your speed is cost effective. It's also fast, right? So and it's, as we know, CTAP is very hazardous. So technicians working with it are exposed to it every day. So what we're trying to do is to also reduce this exposure to all these hazardous reagents. So yeah, again, I can't really answer your question directly because it really depends on how many samples you have a month, per day, or even per year, right? Then you can maybe get in touch with the science work team and we can work this out together with you. Yeah.
Yeah, I think, yeah. So, yeah, okay. Thank you very much for yours response. And also for the first question, I'm trying to understand the questions. I think it's possible to do linkage mapping model for specific dominant genes using F1 population. I suppose that is I and Dusty's understanding about that. And please, Dusty, please kindly respond about that.
So you're making a model in lichen and you want to do linkage map for specific beta carotene genes in an F1 population. That's how I'm interpreting that question. Realistically, to do linkage mapping, I think doing it in an F1 population would be not advisable. You need to get some, you need to ensure that the traits are fixed in the first place before you can actually identify the correct marker that is linked to the beta carotene in lung. Is it? Find me and ask me the question.
Thank you very much, Dusty. And unfortunately, we are limited by time, and we are approaching the Zuhur time, and I'm afraid I need to close this session. However, the question shall not stop here. Everyone can still address the question to every speakers by email. And with that, I would like to thanks again Professor Barat, Brady, and Dusty for your excellent lectures and insight talks. Please give final applause for them. Alhamdulillah, so I'll turn this session to our nice MC, Dr. Dika, please.
Thank you. So before we end the session, maybe for all the speakers and moderator, please remain on the front. And maybe we invite Prof. Dewi Sukma as an Indonesian Horticulture Society to hand over the token appreciation. And maybe also we invite Dr. Widiwanti Wahyu as a head of study program plan and biotechnology study program to hand over the token appreciation. So maybe the committee can take documentation for the first one for Professor Dara Effendi. Can we give an applause? Okay, and the second one for Wadihan. Yeah, give an applause again.
Thank you very much.
And then for those name Fias, or we can call her Bestie. Yeah, please give an applause. And the last one for our amazing moderators. So also give an applause for our moderators today. Yeah.
Please.
Okay. So thank you very much, Prof. Darda Effendi, Bradihan, Dusty for deliver a very insightful talks this morning until this noon. And also thank you, Dr. Sinto Yuniardi, for moderating this session. Finally, ladies and gentlemen, we reached the last agenda today, but before we close, there's several information that I want to announce. For participants who have registered to receive a certificate, the certificate will be sent directly to the email that you register. And if there are participants who wish to receive a certificate but have not yet completed the registration and payment, please fill out the link provided on the event poster. So I think that's all the announcement, and maybe we can have a documentation session. For maybe a committee can please help to have a documentation for the online participant first, maybe. I think there are 3 slides for online participants, so please, for the online participant, you can open your camera and give your beautiful smile to us. Okay, so committee, please help to get a screenshot for the slide. 1, 2, 3. Okay, and next slide. 2, 3. And the first slide. 1, 2, 3. Okay. Next, the recommendation for the offline participants and how to my cell phone can probably maybe from the front, so everybody can remain on the seat, and my cell phone can take our photo.
Oh, agak susah nggak sih? Susah di sini aja. Ini aja ya. Di dari depan. Ini kita di tengah. Belum diatur. Ya mungkin ya kalau ke sini ya. Ya kan sini ya. Atau di belakang sana lebih. Enggak, kita dari depan aja ngambil ke arah sana. Oh, iya. Tunggu di kapan nggak diatur.
Oh, berarti ke depan semua. Dari semuanya duduk aja.
Ya, duduk aja ngambilnya dari sana. Duduk ambil.
So maybe Marisa, can you take a picture from the front? Marisa, dari depan. So everyone, please remain seated on your seat. The committee will take our photo from the front, and please give your cute smile. 1, 2, 3. Cheese. Okay, maybe once again, NGS. Okay, enough. Once more. Okay, please give your freestyle. Okay, thank you. Please give a applause for ourselves. Okay. I think we have now reached the end of our program, and it's time for me to express our deepest gratitude and appreciation to all of those who have contributed to making this event possible and memorable. First and foremost, we would like to extend our heartfelt thanks to our distinguished guests. And we also express our appreciation for the plant breeding and biotechnology study program, Department of Agronomy and Horticulture, and also for the Indonesian Horticulture Society for organizing this program, and also to PT Science Work and LGC Biosearch Technology for supporting this event. Secondly, our sincere gratitude goes to our esteemed speakers, Prof. Darda Effendi, Berkaitan, and also Desti. Thank you for your insightful materials this afternoon. And also, we would like to acknowledge and thank our dedicated helpers and volunteers whose effort behind the scene have ensured the smooth execution of this event. And also, last but certainly not least, a big thank you to our dear audience, both in person, offline, and online participants. Your presence has made this event more engaging and meaningful, and we hope you found it inspiring and also enjoyable, just as we have enjoyed hosting it, and may the insight and experience gained today stay with you and bring the benefit to all of us. Okay, on behalf of the organizing committee, I sincerely apologize for any shortcomings in presenting this event. Once again, thank you for being part of this event. Good afternoon and see you next time. Assalamualaikum warahmatullahi wabarakatuh.